Is there any other questions, or I'm just going to start with Pack 10C? Oh, okay. So I'm going to talk to you now more about the analysis of network topology. We're going to focus on models for generating networks. And we're going to first talk about two different things, what's called ER models and small world networks. So first, the ER model. This is probably the original network model. It was developed by this very famous mathematician named Erdos. Maybe you've heard of this term, the Erdos number, and this is related to his network studies. That's how many links you are away from this famous mathematician in co-publication. And he developed this model, which we're going to talk about. And then we're going to talk about two other models, the Watt-Strogatz model and then the Barabasi-Albert model. Both of these models come from right around 2000. So the ER model is pretty simple, but it's sort of the background that we use to talk about these things. So here we imagine we start with a bunch of vertices which don't have edges, and then we connect a pair of vertices with a certain probability. And the probability, it's called P sub ER, we can vary it. And then one of the things that he noticed is that as you vary that probability, the sort of properties of the graph kind of appear very suddenly at a particular value of PAR, which is around 1 over the size of the number of nodes. And under that, there's really just isolated bits, and then a graph starts to emerge. Now, we'll come back to that probability for PER. That's a sort of random network. But I think the way to think about this is to compare it with other models. So let's talk about a Watt-Strogatz model. And this is a very, very similar model. So in this model, what you imagine is you sort of take the ER graph, which is essentially a random network, and you have on the other extreme a kind of completely regular lattice-like network. So now if we start with the lattice-like network and now we rewire it, we grab an edge with a particular probability and connect it to another vertex, okay? And then we keep doing this with a given probability. Obviously, if the probability is pretty large, we're going to essentially end up with the random ER-like graph. And if it's very small, we're going to have a very regular lattice. But what Watts and Strogatz observed around 2000 was that in the middle, you have this kind of interesting graph, which is you might call it a small world network. And to think about what this small world network thinks, it's useful to think about some properties of a regular lattice first. So regular lattice, all the neighbors in a regular lattice are connected very well, but there's very little long-range connectivity. So that what we'd say using the terminology from last class is we'd say we have a high clustering coefficient, but there's a long path length. Whereas in a random network, the clustering coefficient is not nearly so high, but things are actually pretty globally well connected, and you have a low characteristic path length. And one of the interesting things that Watts and Strogatz observed is that for intermediate values of this rewiring thing, you preserve your clustering coefficient, so the network remains clustered, but suddenly you start to create these short paths through the network, and that's why the network is described as a small world network. And this is actually thought to sort of represent a little bit of what we sort of see in the world, where there's little communities, but then they're randomly connected together, and that leads to overall fairly small path lengths in the whole network. And when they talked about this, they looked at some of these famous examples of networks, the network of film actors, the power grid network, nervous network, and so forth. And they looked at the clustering coefficient and the shortest path in the network. So the small world network is this essentially a thing that has a relatively large clustering coefficient and a relatively small characteristic path. So those are sort of two things. And I'm going to talk about a third type of network, and this is the Barbasi-Albert model, or a scale-free network. Now, the ER network or the Watt-Strogass model, they have a characteristic scale. So if you think about that probability of in the ER diagram, if you think about how many connections things are going to have, it's going to be kind of sort of distributed around that sort of central value. So the PER kind of talks about the average degree of the amount of connections you're going to have. And then, you can have some nodes that have more and some that are less, but it's going to be kind of distributed around that. And that value is a kind of characteristic scale for the network. So on average, you have this amount of connectivity. And the Watt-Strogass model has a similar type of scale, and the scale is the kind of characteristic degree or connectivity that you have. And Barbasi and Albert looked at a somewhat different type of network in around 2000. They looked at what's called a scale-free network. And here, the degree distribution, that is the number of nodes with a particular degree k, that doesn't have any characteristic peak. It just sort of falls off. And if you plot it on a log scale here, that fall off is a straight line. And this type of thing is often, people use the term a power law model. And so the degree, is related to the sort of in relation to this formula. It falls off with the sort of degree to the minus gamma power. And they found that, many commonsensical networks had this property. The World Wide Web, these power grids and so forth, had this scale-free property. And the key idea here is there's no peak at a particular degree. That just kind of falls off. So there's no particular scale that you could immediately see fits this network. And here's, this is a picture from Scientific American that kind of illustrates this. Again, here's a sort of random ER graph network, and which is a typical node. And here's the power law one, where there's no more of a sense of a typical node. It just kind of falls off. And one of the nice comparisons people have for this is kind of the road network, which is to some degree a random network in the United States. It's just connectivity of different cities, but there's a kind of typical connectivity that you tend to have. And then there's what's called the airlines network. And the airlines network, as we'll talk about, grew in a very different way than the road network. I mean, there's some cities that really have a disproportionate amount of flights going from them. They're hubs, and we'll talk about this in a second. And this leads to this power law type of behavior, and it looks a little bit different. Now, so one of the ways of getting a sense of the scale-free network is to think about kind of a growth model for networks. So in the ER graph, we just kind of connected up things with a given probability, and that was fairly uniform. Now in the Barbasi-Albert model, we're going to introduce this notion of preferential attachment. And so what we're going to have is we're going to have a number of vertices, and then at each kind of time step, we're going to add to them. But the probability that we add is going to be proportional to the degree of the vertex. And so what this means is this is essentially the rich get richer type of thing. So you're going to add, the probability of adding is going to be in proportion to how many connections you already have. So if just luckily a certain node gets a few connections, it's going to keep getting more connections too, because you're going to preferentially add to that node. And here's a picture from Scientific American that kind of shows this network growing. You're starting with 2, and then you add something, and notice how you're adding on more and more to this central node, which is becoming a bit of a hub and so forth. And preferentially, not exclusively, not all the time, but preferentially you're adding onto that network. And then there's a sort of way that sort of naturally you can see how this type of thing fits with a sort of gene duplication model. So if you imagine you have two proteins that they're interacting, okay, but imagine that they are duplicated. When you duplicate one node, it will tend to, or one protein, it will tend to go back and interact with its original partners. So that's a kind of type of preferential attachment. And you can sort of see a hub network that has a lot of nodes. If you tend to just uniformly duplicate, it's more likely you'll duplicate on one of the things that already has a lot of connections that will then tend to interact back with that central node, and that node will kind of get, how should I say, more connections, rich get richer in a sense. And so this preferential attachment mode, it actually fits very well with a biological context, where in relation to gene duplication provides a very simple way to see how such a thing, that type of network might grow. And that might explain why people had observed that the protein interaction network has this scale-free topology. Okay, so now I've finished with PAC 10C. Are there any questions there? Anyone wants to ask? So any questions on the ER model, the scale-free network, or the Watt-Strogatz, or anything like that? Okay, so now we're just going to go on to the next bit of network topology. And this will be the final bit. And what we're going to do here is we're going to focus on a corollary of the scale-free model, and that's this notion of hubs, and then which acts as kind of points of centrality in the network. And then we're going to talk about other ways of calculating centrality in networks, and this is a very useful concept. And so we're going to finish up with networks by talking about that with a little bit more biological connection. So first we'll talk about hubs. So again, in the ER network, the degree distribution, that's the distribution of nodes with a given degree, okay, follows this power law formula, and it, on a log graph, it will be straight. And then obviously what that means is there's a few things down here in the degree distribution that really have a lot of connections. And those are, of course, hubs in your network. And one of the interesting things about this is these hubs really become special places in the network, and they dictate the structure of the network. And I think that becomes a way of understanding the network structure. So one thing right away you can see is the network has a kind of different type of robustness and fragility if it's a scale-free network versus a random network. So you can see that because the connectivity of the scale-free network is really dictated by a few hubs, just a very few, it's actually very robust to random attack. It's much more, and random attack means kind of like knocking out a node or knocking out a connection. So if you take your random network and you kind of attack it and you knock out pieces of it, it'll break apart pretty quickly. And by break apart means, for instance, if this was a road network, you'll no longer be able to, say, drive across the United States, because you'll have broken, if you randomly attack this network, many of the connections you would need to make a path across the United States. Whereas if you take a look at the airline network, if you just randomly attack this network, you'll still be able pretty easily to get across the United States. And that's because only a few nodes, which are improbable to be attacked, because there's only a few of them, are really dictating the structure of the network. So the scale-free network is actually very robust to accidental node failure. However, of course, the converse is that it's extremely vulnerable to attack on the node. So if you look at the scale-free network and you say, oh, I want to disrupt this network, it's obvious that if you just attack these nodes with just a sort of a small number of attacks, you'll completely destroy the network with a very small number of sort of node failures. And so it kind of changes the robustness profile of the network. Now, this is very important in terms of biological networks, because what people did was they looked at protein-protein interaction networks and regulatory networks and so forth, and they found that they often had a scale-free topology. And then what they were able to do is they were able to correlate the essentiality, or how important to an organism a given gene was in these networks, to its hubbiness. And basically, what they found in many different contexts was that hubs tend to be essential. And maybe that makes sense, but it really makes sense if you think about the what I just told you about the robustness to attack. So a scale-free topology actually is a very robust framework for protecting your network, because you can have all sorts of changes and attacks on your network, and it won't mess up the essential connectivity, except of course for your central nodes, and they have to be very protected. And one way of saying they're very protected is they're very essential in a biological context. And actually, what one things people have found is that the hubs in scale-free biological networks, such as protein interaction networks or regulatory networks, they tend to they tend to be more conserved. They tend to, the proteins tend to evolve more slowly. There's many measures of how fast things evolve for, like, DNDS would be one particular measure, or there's population genetic things. And in many different contexts, regulatory networks, microRNA networks, protein interaction networks, whatever, people have found that the hubs tend to be more conserved. And here's a picture actually of the human protein-protein interaction network. And what we have here is the more rapidly evolving things are colored in red. And just the way this placement algorithm works, the things on the outside tend to be less likely to be hubs. And the overall picture you have is, I like to say that connectivity is associated with constraint. So the more connectivity you have on a node, the more constraint, the more conservation you have for that particular node. Okay, so that's looking at hubs. Let's look at other measures of centrality. So another measure of centrality is the notion of bottlenecks. So remember, hubs are essentially found from looking at the degree distribution, okay? There's another measure that you can look at, which is the betweenness of a node. And the betweenness of a node speaks to how many of the shortest paths, remember we talked about shortest paths, how many shortest paths in the network go through that node. So if you look at all the shortest paths, say the shortest path between this and this, this and this, and then you look at the path they traverse and you count how many shortest paths go through every particular node, a node with high betweenness or a bottleneck will have many shortest paths going through them. And people think of this metric as a measure of information flow. And I think a good way of getting intuition about bottlenecks and betweenness is think about the road network on the East Coast of the United States, where we live. If you've ever driven through this network, you realize that there's only a limited number of ways of crossing some of the big rivers. In particular, one of the big rivers on the East Coast is the Hudson River. This is a picture of the Hudson River. And there's only a relatively limited number of places you can cross the Hudson River. And one of the most prominent is this structure here, which is called the George Washington Bridge. And so this acts as a bottleneck for all the cars or information packets traveling up and down the East Coast. They have to make their shortest path from wherever they want to go, Boston to Washington, New York to Baltimore, whatever, they have to go through this, these nodes and edges, and that makes this thing a bottleneck. Now, I should point out a bottleneck can also be a hub. It doesn't have to be a hub. You can imagine here, for instance, here's a bottleneck node that is a hub. Here's one that's not a hub. And likewise, you can have a hub that's not a bottleneck, and conversely. Biologically, people have often found bottlenecks bridging disparate genomic processes. So if you have two processes here, often they're connected in some regulatory or interaction sense through a bottleneck edge. And I'm not going to describe it in detail, but there's a particular algorithm that people use to calculate betweenness. It's called the Dijkstra algorithm. It's a very famous graph algorithm that uses dynamic programming, and this is what you would use to calculate betweenness and bottlenecks. So another measure of centrality, let's continue going on, is hierarchy, or tops of hierarchy. And let's start out here with a intuitive notion. So this is a picture of the org chart of Apple Computer from years ago. I don't know if people remember this. This is when Steve Jobs was the head of Apple Computer, and he was the tyrant at the top of this organization. And here's all the people that reported to him, including here's Tim Cook, the current CEO, and then all the other people. And the key idea is that this directed organizational chart really gave you a sense of, it's sort of like a hub, but it's a different type of thing, that someone sits at the top of a regulatory network. And that really points to the importance of that person. Now, one thing we might want to do in a biological network is we might want to find the equivalent of the Steve Jobs, in the regulatory network that's really regulating a lot of things. And how might we systematically do that? Well, if we have a directed network, remember, we can measure what's called the out degree of a node. That's the number of things it regulates, versus the in degree. That's the number of things that regulate it. And obviously, things that sit at the top of a hierarchy are going to have very low in degree and progressively higher out degree. And then obviously things that sit at the bottom of the hierarchy will essentially have just in degree, but not out degree. And so we can kind of look at the difference between these two quantities and think about placing things on a hierarchy in relation to that difference. And there's a variety of different ways of doing this. Here's a non-hierarchy directed network, and here you can imagine how you might sort of put it into a hierarchy. You'd find all the terminal nodes, the things that just have in degree, you put them at the bottom. You then, in a kind of breadth first way, you'd find all the things that connect to these or regulate those, you'd put them on the next level. And then you'd find something that maybe regulates those things and so forth. And so that's one way. Another way of doing this might be some sort of simulated annealing algorithm where you would move things between levels and you would see if it would optimize this hierarchy height type of structure. Now, here's a picture where I actually show, this is the human regulatory network. This is actually in a particular cell. And here is it put into a hierarchy using one of those optimization approaches. And in this approach, all the downward pointing edges are shown in green, but there's a few upward pointing edges shown in red. And what's interesting, I think, about these hierarchy pictures is that they give you a lot of intuition about what's going on regulatorily. Obviously, these things up top are more of the master regulators. These things at the bottom are more the regulated. And what's particularly interesting is that it's almost by necessity, but this mid-level of the hierarchy where there's lots of paths go through, if you calculate the average betweenness of these levels, obviously in the middle of a hierarchy, you're going to get higher betweenness, and you're going to have more bottlenecks or information flow bottlenecks. And I think that's an interesting thing to think about when we think about the structure of networks. Okay, so I think with that, I'm finished with the centrality packet. Are there any questions? Actually, just to make it clear, I had an illustration which I probably didn't explain too carefully of how gene duplication maybe accomplishes this. So imagine we have a biological network that looks like like this, just so we have one node that's connected to one gene, and here's another one that's connected to 4 things. Now imagine these 4 nodes here are, well, they don't have to be, but let's just, they could, let's say they were duplicates of a gene, or let's imagine them duplicating. Okay, so if we said there's going to be a gene, we're going to randomly pick one of these genes that's going to duplicate. So we have gene A, B, all these B's, C, and D. Now we're going to pick randomly one to duplicate. It's much more likely that we pick one of the partners of this guy that duplicates. But remember, when we duplicate a gene, the duplicate's going to want to connect to the thing that its parent has connected to, right? Because it's preformed to connect to that. So if we duplicate this one of the B's, and so we created another B, well, what's that duplicate going to do? It's going to glom back onto A. And again, so it's rich getting richer. Does that explain? Oh, good. Okay. Any other questions? Okay. Let's see how we're doing for time. I think we're doing actually perfectly for time. So we're going to go now to JG, and he's going to present a little bit on eQTL. So I'm going to stop sharing screen and let him take the floor here.
Can you say this? Is it in present demo or what?
I think it looks good. We can see it. We can see it.
Okay, good. Hi, everyone. I'm going to talk about a little bit about the eQTL today, and also I will cover just like 1 or 2 slides for GWAS. One focus in biomedical studies, or specifically in population genetics, is to make connection between the variants and the genes, or the variants to some disease or phenotypes or traits, and also maybe the genes to the traits. So we want to understand the mechanism, what variants or what gene is affecting your phenotypes or diseases. And specifically in population genetics, there are some established analysis or methods that have made such connections. For example, if we want to understand the association between variants and genes, we have the analysis so-called eQTL analysis. Also, if we want to connect the variants to traits, we have genome-wide association studies, or called GWAS. Also, there are also other methods called Mendelian randomization that could use to find the so-called causal genes for some disease. And today I'm gonna focus more on the eQTLs. And to understand the eQTLs, first we need to know what is a QTL, and then we need to know what is actually a quantitative trait. So comparing to the Mendelian traits, that is like the famous example you should have learned in basic genetics, the peas, the color of the peas. It is either yellow or green, and that is controlled by one locus. And yeah, but compared to that Mendelian traits, we have some traits that is more like quantitative. For example, the height of a plant or a human. You see, the height is not a binary values, it's more like a continuous value. And it's sometimes called complex traits compared to the Mendelian traits, also known as simple traits, because these complex traits are usually controlled by a lot of different genes. And you could imagine that if only one gene among all those a lot of genes change a little bit, then the outcome of this, or say the phenotype, might just change a little bit. And that's why you get this like continuous values. So people try to find the genetic locus or variants that is connected or associated with such quantitative traits. and that is called the quantitative tree locus. And this has been done in like 30 years ago, even before the era of sequencing technologies. So in those days, people just find some large section of the genome that are associated with such quantitative traits. And then later on, nowadays, we have the sequencing technologies. So one thing in sequencing technology is the RNA-seq, and we now have the gene expression level of all different genes. And interestingly, we know that this gene expression level, if you look in a population of individuals, it actually has such a similar continuous values, or it follows a distribution. So similarly, we could replace the hive by this gene expression level. So we could ask the question, could we still find some genetic locus that is associated with gene expression? And the answer is yes, and that is exactly what EQTL is. So EQTL stands for expression quantitative trait loci. So basically, that are just genomic locus that explain the variation in the gene expression level. And typically, people divide this EQTL's variance into two groups. One is called cis EQTL, that is the variants are located near the gene, and the other group is called trans, and that means the variant is located distant from the gene, often on a different chromosome. So conceptually, what's going on here is actually very simple. So for example, here in this figure, the x-axis is the genotype, and it is denoted by the number of alternative alleles. So for example, this certain SNP RS1A6 blah, 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 it have the reference allele as G, and then the alternative allele could be A. So if a person, each dot in this plot is a single individual. So if this person have the genotype, both haplotype have G, then you will denote it as 0 in the x-axis. And similarly, if it have both A, then you will denote it as 2. So this is the x-axis, and the y-axis is just the expression level of a certain gene. For example, this in this figure is TMEM253. So after you have all the genotypes and you have the gene expression from all the individuals, you could simply do a linear regression on this. So here, y equals beta times x. And the beta coefficient, you could get a value, and then you could test whether it is equal to 0 or not. And that is the standard t-test in standard linear regression. So this is conceptually very simple. And let's look at some real application or real cases. One of the largest application of EQTL analysis is done by this GTExt consortium. So this GTExt project called Genotype Tissue Expression Project sequenced, they have the whole genome sequencing, whole exome sequencing, and also the RNA sequencing on more than 1,000 people. And they also collect a lot of different tissue from all those individuals. So this is a very large data set. And since they have the genotype, they have the gene expression, they could do this large-scale EQTL analysis. And here's the figure from their data. You see it covers a lot of different tissue types. And in the right-hand side, you see they conduct this EQTL analysis. They have the cis EQTL and also the trans EQTL. Furthermore, they have something called sQTL, but we're not gonna talk about that now. So the GTExt dataset consists of phenotypes, that is a lot of samples times a lot of different genes. And also the genotypes, that is the samples times a lot of different loci, which is the SNPs. And finally, they have a third thing called covariates, basically covariates is some environmental or other non-covariates or variables that might be affecting the gene expression level. So you need to first normalize your gene expression or control it so that it does not have some batch effects or other affected by other factors. So this is kind of like a standard data cleaning process for RNA-seq. So if you are interested in this, you could look at the paper of peer factors, but we are not going to talk about that now. So we focus on the eQTL method that GTEx use, and they use a software called FastQTL. So basically, the following things I would describe is the detail of this algorithm. The first part is the most simple part, that is just the linear regression. They call it a nominal run, and that is to find a candidate QTL for each gene. So you have the dataset that consists of n individuals. You have the expression level for each individual for this one single gene. And you have the genotype. The genotype is basically, you have the n individuals, and then you have, say, L variants or L SNPs. How to find the SNPs? Basically, you just look at this gene, you take like plus minus 1 MB, that is the so-called cis window of this gene. And every SNPs that located within this window will be taken into consideration. And for each of the SNPs, you could do a linear regression, right? So basically, you just conduct the linear regression on this whole matrices. That is this phenotype P equals beta times G, the genotypes. This is just the standard linear regression. And you test whether this beta is equal to 0. You have the nominal p-value for each SNP you test. And among all the SNPs you test, you could find one SNP, let's say 2, that have the most significant p-value. And that SNP Q is considered as your candidate QTL. Any questions so far? Okay, now you take a lot of different SNPs into account and did a lot of different linear regression. So that is a multiple testing problem. You need to control that. So this is the second step, that you want to correct the multiple testing in one gene. And how to do that, you could actually just use a simple like FDR control or family-wise error rate control. But here they did a little bit complicated things, that is to use a permutation. By that, it means, let's say before you have this expression level of a gene, that is this P vector. Now you permute it, just shuffle the order of the expression level, and then you could still do the same linear regression. Before you have this variant Q that have the strongest the most significant p-value, let's say it's called P nominal for this Q. Now using this permuted gene expression level, you could find another variant, Q2, doesn't matter, but it has another most significant p-value, that is so-called p-permute. Let's imagine that if you just have this random thing and you get a p-value that is more significant than your original one, that means your original one is not that rare, or it's very common, because if you just randomly shuffle something, you still get something even significant than that. So the original one will be not rare or will be not significant. And in the other case, if your original nominal p-value is smaller than the permuted p-value, it means your observation is actually very rare. So you should trust this. And that's a simple way that you could use permutation to compare or adjust your original p-value. And here, they didn't use this simple way. They have something more complicated. They found out a mathematical theorem saying that all the statistics could follow a beta distribution. So if you want to look at the kth smallest value from n times of experiments, you have this beta distribution. And basically here in our cases, we look at the p-values from permutations, and we actually looking at the strongest p-value from L different tests. So basically you have this k equals to 1 and n equals to L. And now you have this beta distribution. You could now calculate that if you sample one thing from this beta distribution, what is the probability that this thing you sample is smaller than your observation nominal p-value. And this will give you a new p-value that is so-called beta adjust p-value. So basically the idea is you just need to control the multiple testing problem from your nominal p-value, and they use this kind of complicated beta distribution things. And after that, you get the adjusted p-value, PB. Any questions so far? Okay, now you have one adjust p-value for one single SNP that is connected with one gene. But we wanted to focus on around 20,000 genes in the whole genome. So that's another layer of multiple testing problems. And here they just use a simple FDR control. And in common FDR method controlling methods, there are this so-called BH procedure and also this a more improved version. They just use this improved version, but that really doesn't matter. Basically, it's just a simple FDR control. So with the FDR control, you get the Q value and you apply the filter on the Q value that give you the final significant EQTL associations. That is the genes that is associating with the SNPs. Any questions? Okay, so this is how this GTEST consortium used to calculate or find EQTLs. And you could imagine there are a lot of different applications once you find that, one gene is associated, one SNP is associated with the expression level of a certain gene. That could be very useful. And here I showed a very interesting example I found in the GTEST paper that they first identified a SNP, this RS101, blah, 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 that is significantly affecting the expression level of this gene called IRF1. And here the box plot shows this linear relationship. And later on, they found out that this certain SNPs is actually a trans EQTL that affecting the gene, another gene called PSME1 that is located really far away from this original SNPs. And here it shows the linear regression of this trans EQTL. And similarly, they found out that this single SNP is another trans EQTL for another gene, PARP10. And interestingly, they found out that actually these three genes are within the same biological pathway, and they together control the inflammation response to some certain stimulus. And I think this is a very interesting validation, but also a application how you could use this EQTL association to find out some novel or non-biological pathways, or understand this biological pathway more better. So this is basically how you could associate a SNP to a gene. And the next step is, how could we associate a SNP with a certain traits or phenotypes? And that is the topic for genome-wide association study, or GWAS. And actually, if you imagine that if your trait is actually still continuous, like height, so you could basically apply the almost similar process in EQDL, just replace the gene expression level by with the actual trait values, the height, for example. But and that is actually what people did for those continuous traits. But there's another type of trait, for example, disease, whether you have disease or not. This is kind of binary values. So you could not just apply the linear regression on a binary values. And what they did here is they usually have this case control cohort, and they could calculate the SNPs frequency, or sorry, the allele frequency in these two different groups. For example, if you have disease, you could count how many uppercase A there, small a there, and in the non-disease group, these are two more different values. And you could calculate the so-called odds ratio in this cohort. Here is the odds for allele A, small a, that is basically the number of this small a allele in all the small a's that have disease, divided by those that do not have the disease. So that is the odds for this small a, and here is the odds for the uppercase A. And basically, this odds ratio indicates the effect or the odds for the alternative allele, this small a. If the odds ratio is equal to 1, that means either these two different alleles have equal odds for this disease. If it is larger than 1, then it means if you have this small a, you have an increased odds or increased risk of getting this disease. And it is smaller than 1, that means this small a decrease the risk of getting disease. And with this table, a standard chi-square test could be done to get the p-value for this odds ratio. So basically, in GWAS, if you have a continuous trait, then you could do similar things like EQDLs and get the p-value. And if you have this binary trait, you could do this chi-square test for the odds ratio, and you could still get some p-value. And here, finally, you could plot every single SNPs, the p-value for every single SNPs, and get across the whole genome, and you'll get a plot like this. And people call this Manhattan plot. And here, this dotted line indicates the significant value. So the SNPs among this line will be considered like there are really significant association between the SNPs and the disease you are focusing on. But usually people only focus on the top one, because you see, like here, you have this little line there, then a little gap, and then another little line. That is because a lot of different SNPs are within a one single LD block, or the linkage block, or haplotype block. So basically, they are linked together, and usually people only use the most significant one as a so-called tag SNP. That is the one people always focus on. And basically, that's how people do this analysis for EQTL in GWAS. And thanks. Any questions? Okay.
Well, hold, I think there's a question from Jason in the chat here.
Oh, I see. The question is, does the distribution of a gene's expression in the population matter? It follows a normal distribution, and the model uses standard linear model. However, many genes may be non-normal in their expression across a population. Are non-parametric EQTLs used often? For that question, I think there are some EQTL methods that used a slightly different model, but mostly it's just use the linear regression model. And because mostly the expression level does follow a similar a distribution that is similar to normal distribution, because even though if it is like the Poisson or binomial, if the sample size is large enough and it will finally get very close to a normal distribution. So currently, the most common practice is still using this simple, straightforward weights. But I think there definitely could be some improvement to that. And there's another question in slide 7. In the two heterozygous cases of an EQTL, could there be meaningful difference here in relation to expression? I think this slide. People usually just combine GA and AG together to one single x equals to 1. But there's another type of analysis, so-called allele fold change test or analysis that focus on the different expression level of two different haplotypes if they are, say, having different genotypes. And I think, well, in that case, I think still they combine this GA and HG. So I don't think people really separate these two different cases. And I'm not sure if there are any meaningful differences between them. Okay. Any more questions?
Okay, well, that was a great job, JG. I think that really explained that GWAS and the EQTL really well. I guess any other questions before we'll close class a little bit early today? Any other questions anyone has? Well, we appreciate everyone joining. I guess we'll post the slide packs soon and and stuff. Tinchao and JG, is there anything we want to say about the homework or anything, or not at all? Do we announce anything on that?
Not really. I think, yeah, just look at the homework and it is still next Friday.
Yeah, do your homework. I guess that's the take-home message. Oh, okay. Well, thank you all for joining, and we'll resume in a couple of days, and we'll start the deep learning at that point. Everyone, take care.